as an experimentalist.
Thank you very much for a very nice introduction. So today, I will mainly talk about experimental work combined with theoretical and numerical understandings. So basically, I will talk about bacterial turbulence, as you can see in the background of this movie. And this kind of active turbulence were introduced by Julia in the previous talk. And before starting to talk about science, I guess most of you are worried about or curious about my institution. So I guess most of you have never heard about my university, Institute of Science Tokyo. And actually, this university has just established last year after the merger of two national universities. And I'm basically on the Tokyo Tech side. So my university was actually previously Tokyo Institute of Technology, and now some orders are interchanged, and it's very complicated. So now Institute of Science Tokyo, so abbreviation is Science Tokyo. So this is the take-home message of my talk. So you can forget everything afterwards. So thank you for listening. So I'm kidding. So let me start my science part after showing my collaborators on active turbulence. So these are my collaborators. So my active turbulence, bacterial turbulence study started when I stayed in Eagle's lab during my PhD. So I was a student in the group of Professor Masaki Sano in Japan. And then I stayed in Eagle's lab and started these collaborations, and eventually developed with collaboration with Zabine and Henning and others. And after that, I started my position in assistant professor at Takeuchi Lab, and we continue collaborations like that. And then this is just a brief introduction about myself, because there are many people that I meet for the first time. So I've been working on both artificial, non-living Janus particle systems driven by AC electric field, and also working on bacteria, and almost all of my work are on non-intelligent, simple active matter systems to understand basic physics behind active matter systems. And we've been studying fragile dynamics of the Janus particles at the single cell levels, single particle dynamics, and also the macroscopic dynamics. And also we are studying the cell motility and swimmings, and also the macroscopic bacterial collective motion. And today I will focus on this turbulence, macroscopic behaviors. And as Julia introduced in the previous talk, we often see turbulent behavior at the microscopic Reynolds number regime in active matter systems. So, and these are the examples from some of the experiments, like bacterial systems, Janus particles, and epithelial cells, and cytoskeleton systems. And as you already know, so in the case of bacterial turbulence, so each bacteria are swimming at the low Reynolds number regimes. But due to the active stress induced by each bacterium, the additional non-linearity appears, and as a result, we observe non-linear dynamics. And the basic mechanism of the turbulence is like this. So due to the steric interaction between the bacteria, they tend to align like pneumatic liquid crystals. But due to the activity, they push each other through the hydrodynamics or mechanical contact, and then the thread configuration becomes unstable, and then the continuous buckling leads to the turbulence-like behavior at the microscopic scale. And this kind of bacterial turbulence has been well reproduced by using these kind of hydrodynamic equations called Tonatu-Swift-Weinberg equations, in which can be derived phenomenologically by implementing active stress term, self-propagation term, and vortex formation term into the Navier-Stokes-like equation. And the important thing here is that due to this active stress term appears as a coefficient of the advective term, and this coefficient becomes larger than 1 in the case of pusher-type microswimmer systems, like bacteria. And so here the point is that in these equations, we only consider about the bacterial velocity field because in standard microscopy, we don't see fluid velocity. So in this equation, we only deal with the bacterial velocity field of their bodies. And by using these hydrodynamic equations, people have succeeded in reproducing macroscopic behavior of the bacterial turbulence, as you can see in these numerical simulations. And we can reproduce, for example, some statistical properties, bulk statistical properties like power spectrum of the velocity. But then the question is, what happened in the presence of the boundaries? And the interesting thing about this active turbulence system is that all of these systems have characteristic length scale with the vortices, as you can see in these schematic images. And as a result, once they are confined in circular geometries, they tend to self-organize into a stabilized vortex structures like this. And then our question is like this. So the hydrodynamic equations for the bulk active turbulence has been proposed. And then how can we deal with the boundary condition in these confined systems? And to understand these questions, we previously worked on this kind of experiments with Igor. So we constructed microscopic pillars like this on the top glass substrate, and we put bacterial suspension of Bacillus subtilis, and then we observed at the liquid air interface. And the reason why we observe liquid air interface is that bacteria, Bacillus subtilis, is an aerobic bacteria, and they like oxygen, so they try to accumulate at the liquid air interface. And as a result, we can observe effectively two-dimensional dynamics, and the bacteria becomes very active only at the liquid air interface. So we can observe effective two-dimensional dynamics, and in addition, due to the sufficient supply of the oxygen, we can observe bacterial dynamics relatively for a long time. And then we construct microscopic pillar arrays with periodic microscopic pillar arrays with different periodicities, and we observe how bacterial bulk active turbulence flows into these structures and how they behave inside these structures. And after analyzing the velocity field and calculating the vorticity field, so we obtain the time average vorticity field like this. And as you can see here, in appropriate length scales, red and blues appears alternatively within the lattice of the microscopic pillars. And if you enlarge this area, it looks like this. So red and blues represents the directional rotations. So within the pillars, among the pillars, each area counter-rotates. So that we see counter-rotating neighboring vortices like this. And so we observed antiferromagnetic vortex order inside these lattice structures. And the most stabilized vortex order was achieved at a lattice constant about 70 micrometers, which actually corresponds to the bulk bacterial turbulence, bulk creation length of the bacterial turbulence. And this lattice size is quite small, 3 times 3 for statistics. It's very small for statistical physicists. So we try various sizes of the lattices, and we evaluated the order parameter by changing the lattice size, and we concluded that even for system size goes to infinity limit, the vortex order can span the whole lattice without any defect. And this is the largest lattice we constructed, and as you can see, the anti-harmonic vortex order spans the whole lattice. And then we continued to ask what kind of lattice can host antiferromagnetic vortex order. And as a first trial, this is a very simple experiment. So we constructed honeycomb lattice, which should have fluctuations if we try to make antiferromagnetic vortex order. And as a result, we couldn't realize global antiferromagnetic order as we expected. We only see some local order. And then the question is, what kind of lattice can stabilize vortex order? And in order to answer this question, we look at this flow field in a topological way. So if we write down the flow field schematically, it looks like this. And then we noticed that the pillar can be regarded as a -1 topological defect with a velocity field. And then, but you may think that, so this is a kind of trivial because they are aligned in a regular lattice. So we try to observe an isolated pillar and see how it behaves. So we extracted one pillar, we isolated one pillar and observed the velocity field around them. And then still, we found that the isolated pillar can behave as a -1 topological defect, meaning that the topological defect, -1 defects, can be trapped by an pillar by itself. And for the moment, our understanding about the antiferromagnetic vortex order formation is the following. In the bulk, plus and -1 topological defects are flowing around, but as soon as we construct microscopic pillars, then -1 topological defects are selectively trapped by the pillars, and as a result, we observe antiferromagnetic vortex order like this. And then we questioned how we can predict their behavior in the presence of boundaries. So we looked at the boundaries carefully and looked at the experimental data, and we identified we inferred experimentally the boundary conditions like this. So not only the velocity field goes to 0 at the boundaries, but also the vorticity field seems to go to 0 at the surface of the pillars. So then we try to implement this boundary condition into the bulk bacterial turbulence equations, as I already introduced in the introduction part. And here, due to the higher order derivative term existing in these Tonatu-Swift-Hohenberg equations, we need additional boundary conditions compared with Navier-Stokes equations. And we implemented these boundary conditions to see whether we could reproduce the macroscopic vortex order as well as the topological flow topology around the pillar, around the pillar and the pillar lattice. And today I'm skipping, but by changing the pillar diameter, we can also control the topological winding number of the flow field around the pillar like this. So as we increase the diameter, the -2 and -3 topological defects start to appear. So these are the experimental data points. And our question was, so whether we can reproduce these experimental results or not. And the question was, of course, yes. So by implementing these inferred boundary conditions, we managed to reproduce macroscopic vortex order and also the topological flow topology around a single pillar analytically, and also this dashed line is the numerical results, and this numerics quantitatively reproduced experimental observations. And after we managed to, after we established this numerical scheme, then we can use this scheme to predict and design our experiments. And for example, by arranging the -1 topological defect in a Kagome lattice manner, then we can predict that the anti-vortex order can also be stabilized in the Kagome lattice like this. And the interesting thing about Kagome lattice is that depending on the initial conditions, we have two kinds of realizations. So in Kagome lattice, in this case, the larger area, the larger hexagons are rotating in the clockwise, and the smaller triangles are rotating in the counterclockwise direction. But there's another realization where the hexagon rotates in the counterclockwise direction. So this order is realized after the spontaneous chiral symmetry breaking, and as a result, we can predict that there is a non-zero circulation around the lattice structures. So it can be used as an active fluidic devices, and then after this is just a numerical predictions and theoretical predictions. So based on this prediction, we can of course fabricate the microscopic structures and see whether we can stabilize a vortex order. And indeed, we could stabilize antiferromagnetic vortex order like this, where the hexagons have clockwise directions, clockwise rotations. So with this method, we can now predict microbial collective motion in the presence of boundaries by using partial differential equations quite precisely. And then we start to apply this method to understand more fundamental questions on bacterial turbulence and active turbulence. So far, what we have been looking at is how this spatial-temporal chaotic behavior is stabilized under the boundaries. And then our question is the opposite. So how do these vortex orders get destabilized? So if we classify these three systems, so they have different symmetries, actually. So in the case of epithelial cell sheets and cytoskeletons, they have nematic interactions and nematic alignment. And in these systems, particularly in the reconstituted cytoskeleton systems, the route to active turbulence has been quite well characterized, and the understanding is like this. So as you see, at some point, plus and minus 1/2 topological defects are created, and they start to move in a chaotic motion. And this unbinding of plus and minus 1/2 defect destabilizes the vortex order and eventually leads to active turbulence. And then our question is what happened in the case of bacterial turbulence in which the bacteria aligns in a polar way? So this is our question. So in other words, it can be rephrased like this. So how the route to bacterial turbulence looks like under the circular confinement. So in a small confinement, we know that bacteria self-organize into a stabilized vortex, but as we increase the radius of the confinement, it should eventually reach to the bulk bacterial turbulence. So what's the intermediate state, and what is the route to the active turbulence under confinement? This is the question. To understand these questions, we fabricated a microscopic arrays of cylindrical wells, and in order to analyze and evaluate the stability of the vortices, we fabricated the whole device with some PDMS thin membranes, which are highly oxygen permeable, so that the bacteria can keep high motility for a relatively long time. And this is our setup. So the wells aligned in the vertical line have the same radius, and it increases by 0.5 micrometer step in the horizontal alignment. And we prepared about 400 wells in a single field of view to analyze the transition. And because at the transition point, usually the dynamics is quite fluctuating, so we need to prepare a lot of wells to analyze the dynamics by using high statistics. And this is the typical snapshot of the vorticity field. And as you can see, in a small enough radius, we only see one vortex fits inside the well. But as soon as we increase the radius, we start to see more vortices inside the well. And these are the So these are the typical snapshots of the vortices. So in a small enough radius, we only see one vortex. But as soon as we increase the radius by 1 or 2 microns, we start to see 2 vortices and then 4 vortices. And we observed the subtle change in the radius yields transition from 1 and 2, 4 vortex states. And the dynamics looks like this. And for one vortex state, the direction rotation is stabilized and it continuously rotates in the same direction. But for the 2 vortex states, the 2 vortices start to compete with each other, and the direction rotation starts to reverse rather periodically. And then in the 4 vortex states, the 4 vortex start to pulsate like this. And then we try to characterize these transitions. So let's first look at the first transition from 1 vortex state to the 2 vortex state. And to characterize the state, we define the spin variable inside the well. So this is just the angular momentum within the well. And this is obvious, but for steady vortex state, the spin variable stays in the same sign, while in the reversing vortex state, it reverses alternatively like this. And we define the correlation time from these spin variables. And then we made a scatter plot of the correlation time as a function of the radius. And this red line is the moving median of the correlation time. And for small enough radius, it doesn't change the sign during the period of the observation. while as we increase the radius, the correlation time becomes smaller and smaller, and we can observe that the transition from steady vortex to unsteady vortex like this. And one important thing here is that if we decrease the radius from above, this means that the period of the reversal start to diverge at the transition point. And this is actually consistent with our theoretical analysis that I will present later, in which we show that the transition is infinite period bifurcation. And then if we look at, again, how the vortex reversal looks like, and then we notice that this reversal is rather regular and periodic. And if we make the histogram of these spin variables, then we see bimodal distribution, meaning that there's two relatively stable configurations, clockwise and counterclockwise rotations, and the system hops between these two states rather periodically. And then we start to characterize the pulsating state of the four vortices. And in order to characterize this four vortex state, we apply Fourier mode analysis. So we decompose the velocity field into the azimuthal Fourier modes like this. So the lowest three modes are depicted here. So we decompose the velocity field into the one vortex mode, two vortex mode, and four vortex mode, and so on. And then we calculate the kinetic energy of each mode, like this, defined like this. And then we plotted the kinetic energy of the two vortex mode and four vortex mode for the pulsating states. And then we noticed that these two modes have peaks alternatively like this. And if you look at the snapshot of the peaks, indeed, we observe that the four vortex state and two vortex state appears alternatively like this. So this anti-phase oscillation of the two vortex mode and four vortex modes seems to be responsible for the pulsation of the four vortex state. So this is a quick summary of our experimental observations. We observe one vortex state stabilized for a long time, and then we start to observe regular reversals, and then four vortex pulsations. And then we wanted to explain and describe these transitions numerically and analytically. And we carefully examined which hydrodynamic equation is the best to describe our experiment. So I already introduced about our previous work with the pillar lattices, and we used Tonatu-Swift-Holmberg equations, and we successfully reproduced the experimental results. But that experiment was done at liquid air interface, while our current experiments were done between two solid plates. So in order to be careful enough to choose which hydrodynamic equations to use, we compared two equations. The first is the Tonatu-Swift-Holmberg equations, as I explained previously. And the second one is the so-called Nikolaevsky equations, as you can see here. And these two equations have different instability spectrum, as you can see here. And we tried two equations to see which equations fit better our experimental data. And to see this, we applied linear regulations of these linear equations. We applied our experimental. We applied linear regression on this experimental data to these two equations. And to avoid the boundary effect, we only use the data away from the boundaries. And as a result, we obtain that the Tonatu-Swift-Holmberg fits always better than Nikolaevsky equations. So this is a result of the residual from the linear regression analysis. And the Tonatu-Swift-Holmberg equation always has smaller residuals than Nikolaevsky equations. So we decided to use Tonatu-Swift-Holmberg equations for our experiment between two solid plates. And then we can safely apply our previously established numerical scheme on our current setup. And then we apply this setup, numerical scheme, and then we managed to reproduce our experimental observation of single stabilized vortex, reversing two vortex states, and four vortex pulsating states. And if you look carefully at these reversing vortex states, and then we calculate the spin variables also for the simulations, then we observe perfectly periodic orbits in our numerical simulations. So we can conclude that this reversing state has periodic, is a limit cycle oscillations. And then we try to understand what the mechanism behind these oscillations. And to understand this, we again apply Fourier mode decomposition of this original velocity field, and we decompose them into the one vortex mode, two vortex mode, and three vortex mode, and so on. But now we realize that we can only use the lowest three modes because the lowest three modes have higher amplitudes than the others. And by using these three modes, we can almost perfectly reconstruct the original velocity fields. So we only focus on the first three modes, and then we try to find the analytical expressions. And these theories are developed by Igor, and I will briefly explain about the theoretical details. So we start from the vorticity representation of the Tonatu-Swift equations, and then we linearize these equations, and then we can find the general solution of these linear equations. And the solution can be described by using the mode omega n with the growth rate lambda n with the Bessel functions and characteristic wave numbers. And then if we impose boundary conditions at the, where v and omega becomes 0 at the boundaries, then we can obtain the specific solution for our problem with growth rate and wave numbers. And if we compare our linear solutions with our PD solutions and experimental results, so these linear solutions can almost perfectly explain our observations. So this is the numerical comparison between numerics and linear solutions. But without any fitting, the linear solution almost perfectly explains the PD solutions. And this is the fit of the linear solutions to the experimental data. But the linear solution can also, again, perfectly explain our experimental data. And then we decided to use this linear expression. And then what we did was a weakly nonlinear analysis. So we expand the original vorticity field by using the eigenfunction of the linear solution, linear equations, like this. And then we plug this representation into the original nonlinear partial differential equation. And then we obtain the ODE equation like this. And here, the C is the real values, and the A1 and A2 are complex variables. So we obtain 5-dimensional ordinary differential equations. And then we numerically integrate these equations and obtain the limit cycle oscillations like this from the analytical calculation. So and we found that this limit cycle has the bifurcation of the infinite period bifurcation, and this is consistent with our experimental observation with diverging period of the reversals. And this analysis not only reproduces the limit cycle, but the trajectory in the phase space is almost the same as the numerical results from the partial differential equations. And as we increase the radius, of course, the analytical results can also reproduce the pulsating four vortex states, as you can see here. So as a result, we identified a route to active tolerance via oscillatory states in experiments, numerics, and analytical results like this. And then the final question is, what's the mechanism of the oscillatory behavior? And this is our understanding. So if we look at the amplitude of the lowest three modes, all the three modes has comparable amplitudes. So all the three modes seems to be important. And from our analysis, we know the linear growth rate of each mode. And as soon as the zeroth mode, which is the one vortex mode, becomes unstable, we start to see corrective rotation of the stabilized vortex. But as soon as the the two-vortex mode, n equals to 1, becomes unstable, we start to observe oscillating two vortices with the finite amplitude of the second mode, the four-vortex mode. Even though the n equal to four-vortex mode has negative linear growth rate, meaning that this coefficient is negative and A2 is linearly stable. But the reason why we observe positive finite amplitude of the first mode is that we have nonlinear couplings between the zeroth mode and first mode, and this excites the four-vortex mode. And actually, these coefficients are proportional. These nonlinear couplings are proportional to the lambda in the nonlinear advective term. And this nonlinear advective term actually originates from the active stress term. Active stress generated by bacteria. So this reversal can be regarded as generated by the active stress generated by bacteria. And indeed, if we only observe limit cycle when the lambda is larger than 3.7 in our analytical calculations. And the regression of our experimental data to the Tonat-Swiss-Hohenberg equation reproduced that the lambda is about 4.2, which is again inconsistent with our analytical calculations. Okay, so this is the summary. So we identified that the reversal is a precursor of active turbulence, and we observed reversal both in experiments, numerics, and analytical calculations. And we also identified the bifurcation, and bifurcation should be robust. So we believe that the orchestration of these three lowest modes is the generic for the onset of polar active turbulence, and we anticipate that this mechanism can be applicable to other active systems, other active polar systems, and other geometry, and so on. So we look forward to test this understanding in other systems and so on. Okay, so that's it, and thank you for listening, and I'm happy to answer questions. Thank you very much.